think that the major advantage that we will see in the next years is that we can discover something fundamental about the human-to-human variability. I'm Prisca Liberali. I'm a professor at ETH Zurich at the Department of Biological System and Engineering, and I'm co-affiliated with the Friedrich Miescher Institute in Basel. So intestinal organoids are 3D structures that mimic the intestine in vitro. They can grow from single individual stem cells from a healthy individual and patient, and you can create the full tissue organization of the intestine with the cell type composition and the spatial organization. And I think what really represents a change is that not only the tissue composition is recapitulated, but also the functionality of the tissue. So we can mimic a lot of the function of the different cells, of the tissue, but also mimic disease. I think the core of self-organization that is really intrinsic of stem cells and and really show how organoids can form is the communication. Both the sensing of its environment, so cells in a way are little units that are able to sense the environment and behave accordingly in space and time. And I think this is really the core principle. It's not only the communication, but it's sensing the environment and communicating with the sensing. And so the cells are able to know where and when they are and respond autonomously to, and I think the other aspect that is very important is also the coordination. Cells are able to understand what the neighbor do and coordinate together. When we try to look systematically at this organoid, combining the live imaging, the genomic, and the modeling is fundamental to understand the complexity of the system, especially if we think about the self-organization, but all developmental and regenerative processes. These are very complex processes that require the integration of spatial method like imaging, more deep method like genomics, and the computation that really can integrate all the methods. And without these, it's very hard to have a full global picture of what is understanding of the basis. And at this point, I think one of the major discovery that was possible is when we developed the first organoid screen, image-based organoid screen. In this screen, we performed around 3,000 compounds. We looked at millions of organoids, and from these organoids we were able to find a drug that is improving regeneration in vivo, directly in mice. And this was only possible because we are not increasing regeneration everywhere, but we were really looking at the window of improving regeneration. And I think this was only possible combining the three different approaches that we have in the lab. I think we have been moving really quite far in developing and bioengineering better organoids. The main issue that I think at the fundamental, we need to understand which aspects are recapitulated by organoid. Some organoid really recapitulate developmental process, some organoid really recapitulate more regeneration, and really understanding this window of what in vitro is recapitulated is very important. Bioengineering has made incredible improvements on adding vasculature, immune component, and really increasing the breadth of what can be done. But I also believe that keeping some animal model will be important to really combine in vitro system and animal model to really get the full picture of what is happening. It's really a tool that increase our capabilities in the lab, but won't solve all our problems. I think that the major advantage that we will see in the next years is that we can discover something fundamental about the human-to-human variability. We have been sequencing humans since many years, and it's 20-25 years, and we still lack some very fundamental understanding of where the causes of some human-to-human variability comes from, sensibility to acquiring some disease, to inflammation. While I think that being able to make organoids directly from individuals. So we have hundreds, and if not thousands, of organoids directly from healthy individuals, from different ages, from different gender, from different races. And understanding this variability, I think, will be something that will be very important for the future, to understand how they react to stress, how they react to inflammation, how they acquire disease, and how they react to drug, and to make drug much more personalized, both on the full population of and the human-to-human variability.